So our focus was on creating an adaptive network model which simulates the role of the gut microbiome on the circadian cycle, which is the 24-hour body clock, mood and insomnia, which is the sleep disorder where you struggle to fall asleep, through the gut-brain axis. So the microbiome is the population of microorganisms that live on your body, but we focus specifically on those in the gastrointestinal tract, so in your gut. And the composition of this microbiome affects different biological processes in your gut. So these can then travel along the microbiome gut-brain axis using neurotransmitters, hormones, or inflammatory cytokines to affect the biochemistry of your brain. This can affect mood, circadian rhythm, which is that 24-hour body clock, and insomnia. So there's three main pathways which are part of this microbiome gut-brain axis. These are the neuroendocrine pathway, the immunoregulatory pathway, and the vagus nerve pathway. But we decided to focus on the neuroendocrine and the immunoregulatory pathways because we wanted to focus on the endocrine and immune systems and how they work to regulate different hormones that are important for a regulated sleep cycle. So we've got our neuroendocrine pathway on the left, and this is where endocrine cells in the gut are stimulated by microorganisms. Depending on what microorganism, they release different neurotransmitters. These then travel along the bloodstream along the HPA axis, which is the hypothalamic pituitary adrenal axis, which coordinates the body's stress response. And this is coordinated through cortisol regulation, because cortisol is the main stress hormone of the body. So tryptophan metabolism is also regulated through this pathway, and tryptophan is a precursor of the hormones serotonin and melatonin, which are both very important for a regulated sleep cycle. And then we have the immunoregulatory pathway, which is the pathway to the right. This is where immune cells in the gut detect pathogenic microorganisms. So it causes these cells to secrete pro-inflammatory cytokines, which travel in the blood as well. And these cause inflammation throughout the body. Sometimes these pathogenic bacteria can produce neurotoxic metabolites, and these then travel through the blood to the brain and can cause damage there to many different mechanisms that affect mood, circadian rhythm, and can increase your chances of insomnia or worsen it. This also can cause inflammation in the gut. So you can get a leaky and inflamed gut from this, which means that these toxins can more easily pass into blood. Next slide, please. So if you want a healthy microbiome to reduce your chances of insomnia and improve it, then you can try to reduce your stress, have a healthy diet, maybe take some probiotics, because lower stress means lower cortisol levels. And when cortisol is too high, this can dysregulate your circadian rhythm. And this also dysregulates the clock genes, which are the ones that encode for proteins that regulate that circadian rhythm. We chose to focus on two probiotics, bifidobacterium and lactobacillus, because these are both found in high concentration in your gut microbiome anyway, because they've got very beneficial effects in helping to upregulate serotonin and melatonin production in your body. And if you have more serotonin and melatonin, generally insomnia is improved. So the current drugs on the market for insomnia have lots of side effects, and they're not very effective over the long term. So research into these probiotics for treating insomnia would be very necessary. Next slide, please.
All right, so I'm going to talk briefly about our states. We've got 30, as you can see on the screen, and we've got lactobacillus, bifidobacterium, and bad bacteria as some of our states. But I'd mostly want to focus on our adaptive states. So we chose to make all of our neurotransmitters adaptive states. So we've got melatonin, cortisol, and serotonin, as Claudia mentioned earlier. And we chose to make those adaptive because we wanted to more accurately reflect the adaptive neural pathways that they follow. So we're changing their connection weights as well as their speeds through second order adaptive networking. Two other aspects that we wanted to change through adaptivity are the external stressor distress connection. There's been a lot of research done that people who undergo external stressors, especially when they're young, tend to have a much higher level of stress response. So we wanted to also reflect that in our model. And we also wanted to show how the immune system can also be adaptive. So we made the connection between bad bacteria presence in the gut and the immune cell response to be adaptive. Here you can see our model for the network. There are, like Claudia mentioned, two main pathways that we focused on. The first is the immune response, which you can see on the right hand of the screen, where the bad bacteria in the gut triggers an immune response, which triggers more inflammation. And that eventually leads to more cortisol presence, which leads to insomnia. And then we have the HPA axis, or the neuroendocrine pathway, triggered by the presence of the probiotics. So lactobacillus, it actually reduces cortisol production, and bifidobacteria increases serotonin production and also increases tryptophan availability, which is necessary to produce melatonin, and both serotonin and melatonin increase the body's ability to regulate clock genes, which then in turn reduces insomnia. On this model, we have four inputs. So the first is the external stressor, which remains constant throughout the entire model. And the other three are the bacteria in the gut, so the lactobacillus, bifidobacteria, and the bad bacteria in the gut. So those are our four inputs. We use four combination functions: the identity function, advanced logistic sum, Hebbian learning, and bounded growth. Everybody knows we use identity for those that take in one input, advanced logistic sum for all the ones that take multiple inputs, Hebbian learning for our W states that affect connection weights, and bounded growth. We use this one for the three bacteria, the lactobacillus, the bifidobacteria, and the bad bacteria, because we wanted to model how bacteria tend to grow in an environment.
Okay, so in the last part of our presentation, we'd like to describe in detail which specific scenarios we decided to model in MATLAB with respect to our domain, and our intended expectations from these simulations that we ran, and our final inferences from the graphs. So in this analysis part, we ignore the first and second order adaptive states just for the purpose of analysis, as these do not actually represent like actual body states in the system of a person, even though of course they're crucial for the purpose of modeling. So in our first scenario, we have low levels of probiotics and high levels of bad bacteria in the gut. So we intended to look at the effects produced on the system's state of insomnia, which is X15, and its effect felt as sleep deprivation, which is the X16 state that we see on the screen. So in the scenario of imbalance of bacteria, such that we have no probiotics, such as the cortisol regulating lactobacillus or the serotonin regulating bifidobacteria present in the gastrointestinal tract, but we do have the bad or pathogenic bacteria, such as Clostridium, perfringens, E. coli, etc., that release toxins and or trigger disease. So this state is what we term dysbiosis, or just the presence of the bad bacteria in the blood. We have achieved this state in MATLAB using the IV values 0 and 0 for LSC and PIF, which is our good bacteria in this scenario, and we use an IV state of 1 for the bad bacteria, and of course the bounded growth function for it. So next slide, please. So if I describe in detail the states that our first scenario achieves. So since the bad bacteria in the gut is high, we get an immune response, which means that our X4 is high, X4 state, which in turn activates the cytokines, which leads to gut permeability or the leaky gut, which means the bad bacteria in the gut then permeate into the bloodstream. And this causes the HPA1 axis, which is just our way of referring to the different modes of activating the HPA pathway. So we have HPA1 and HPA2. So the bad bacteria works on HPA1, and it leads to high levels of cortisol, which means the body perceives the state of stress, which is our X11 state, much more higher. And then we have the other enteroendocrine pathway, that is ECC2 and HPA2, which is regulated by the bifidobacteria that is completely absent, and we do not have a serotonin level because our good bacteria in this scenario are completely absent and zero. This in turn, can we go back to the previous slide? So this in turn, as we see, it causes a high X15 state, which leads to a high X16 state, which is a sleep deprivation, as we can see from the graph, as there is no good bacteria present at all. And since the EEC2 pathway is completely inactive, we do not have any such effects on tryptophan availability, melatonin production, or the clock gene regulation, which is done by the good bacteria. And like we mentioned, our external stressor or the X20 state is 1 throughout our scenario. Now we look at our second scenario, in which we assume high levels of probiotics, but low levels of bad bacteria in the gut. Again, like our previous scenario, we have achieved this by having IV states of 1 for both lactobacillus and bifidobacteria, and a state of 0 for our bad bacteria in the gut, which means that there is no immune response that is triggered in the body, no cytokine response as such. Because of that, there is no gut permeability because there is no bad bacteria. And of course, we do not have a state where this bad bacteria can then permeate into the bloodstream. But because of the presence of lactobacillus, next slide, please, our EEC1 state, which is the first half of the enteroendocrine pathway that affects the HPA axis, that is activated, which causes, which then results in the HPA1 axis to be downregulated, and this causes the cortisol levels to fall, which then causes our body to perceive stress as low, which ultimately, as we know, is regulated by the lactobacillus. Then in the second pathway, where we see the effects of bifidobacteria, our enteroendocrine cells and the resulting HPA2 pathway and the serotonin levels are high, which means that our perceived states of insomnia and sleep deprivation are in turn low. And there is an increase in tryptophan, which actually produces melatonin and serotonin, and this means that our X19 state, or the clock gene regulation that is done by our good bacteria, is working. Now we look at our third scenario, where we have taken all our states as low, which means that we do not have any such bacteria in our gut. We have low levels of probiotics as well as low levels of bad bacteria, which we have achieved by taking our IV states as 0 for all. So we do not have an immune response as such, but since our external stressor is 1 throughout, next slide, please, our cytokine response is high, which then results in high cortisol and stress levels because there is no regulation because of the absence of lactobacillus and bifidobacteria. The gut permeability that is perceived by the body is seen to be high because the leaky gut syndrome is then activated, even though there is no actual bad bacteria in the blood. In terms of the the HPA1 and HPA2 axis, that is irrelevant to our study, as there are no, in this particular scenario, as there are no bacteria in the gut whatsoever, and the tryptophan, melatonin, serotonin productions are all low, and there is no clock gene regulation. So these are the 3 specific scenarios that we decided to simulate. I can understand it's a bit confusing because of so many colors and lines, but I realized that if I actually share from MATLAB, it's going to break the flow of our presentation. So thank you for bearing with me. Now I'll talk about the limitations of our model. So these are basically some ideas, some extra ideas that we had that could result in an extended model, but we actually did not have enough time to implement all of them. So the first one would be the bacteria in competition. So we have negative connections between the bad and good bacteria, which then interact with each other and affect the state of mood, sleep, and other mental states in our body. The second one would be when we tried to do a comparative analysis with previous year's summer group, where they did more real-world practical states, such as when you suffer from jet lag and such. But we decided our approach is more focused on the biological aspects and the levels of bacteria. We also thought about a 1-1-1 state where all the bacteria are active and interacting and present in the gut. We thought about having different speed factors for the external stressor values instead of having it constant throughout. And there is also something called opportunistic bacteria, where instead of just the good and the bad bacteria, we have bacteria that for the most part remains dormant, but then when the immune system is low, it activates and causes the same effects as the pathogenic bacteria. So that would be it for our presentation. Thank you.